Emily Spaulding is an assistant professor and the George Wojtech Endowed Chair in Neurobiology at the MDI Biological Laboratory. She is a Maine native. She uses C. elegans to study the nucleolus and its role in neurodegenerative diseases. And during her postdoc at the MDI Biolab, she was recognized by the National Institutes of Health as an outstanding scholar in neuroscience. Emily, I also roped into a podcast conversation. So we did a deep dive into actually her whole background. Emily is this really cool backstory that I am not going to tell you, but I would encourage you to listen to it. And the best part, Emily has had perhaps one of my most favorite answers for favorite pop culture science item. She doesn't have one. When she's not doing work, she relaxes by listening to the podcast Till the Rafters Ring, which is great.
Maine hockey.
Maine hockey. It's a Maine hockey podcast. So that's where she's spending her time. All yours.
All right. Thank you so much, Kate. The goal of my research is to answer the question, what drives neurodegenerative disease? And the types of disease my lab focuses on are incurable forms of age-related neurodegeneration, such as ALS. In ALS patients, cells in the region of the brain controlling locomotion slowly degenerate, resulting in loss of muscle control and ultimately in death. But how this disease unfolds inside brain cells is largely unclear. Would you believe me if I told you that a tiny worm may actually hold the key to our understanding of devastating neurodegenerative diseases like ALS? Well, despite being obviously quite different in size and shape from human beings, these tiny worms called C. elegans actually share 70% of their DNA with us. And unlike humans, we can peer directly inside the brain cells of these animals while they're still alive because they're fully transparent. And so because of these advantages, C. elegans have been used for decades to uncover biology related to human health and disease. And in fact, research using C. elegans has led to 4 Nobel Prizes and contributed to therapies for cancer. But you still may ask, how could we look to a lowly worm to study a disease of the human brain, which is possibly the most complex object in nature? Well, that's just the challenge. With around 86 billion cells, the human brain is so complex that we still only have a pretty basic understanding of how it works. And even the mouse brain is incredibly complex with 100 million cells. At the other end of the spectrum, another common laboratory organism, yeast, doesn't have a brain for us to study at all. And then we have C. elegans, which in my humble opinion sits right in the sweet spot between simplicity and complexity. Each and every C. elegans has exactly 302 brain cells, no more, no less. And we know quite a bit about each one of those brain cells. For example, we know the gene activity inside each brain cell, the circuitry of each cell, and the function of almost except every one of those 302 brain cells. So because we know so much about the healthy C. elegans brain, it's a nice starting place from which to understand what goes wrong in disease. And my lab focuses on an essential compartment inside brain cells called the nucleolus. In brain cells of healthy individuals, the nucleolus is intricately organized into multiple layers. But in brain cells of ALS patients, this layered organization is disrupted. However, whether this disrupted nucleolar organization contributes to the disease is currently unknown. So to study this using C. elegans, we first edit the worm's DNA to introduce fluorescent tags that will label the nucleolus inside brain cells. At the same time, in some worms, we introduce a mutation in a nucleolar component that will cause nucleolar disorganization. We do this by micro-injecting DNA editing tools directly into living worms using a tiny needle under a simple light microscope. We next use a high-resolution microscope to visualize the tiny nucleolus inside brain cells of living worms. And using these approaches, we can see one nucleolus, which is comprised of 2 distinct layers in a healthy worm, and then how this layering is disrupted in the brain cell of a worm with the nucleolar mutation. But does this nucleolar disorganization cause ALS-like symptoms? To answer this question, we take videos of freely moving worms and quantify their locomotion. And we find that compared to healthy worms with an organized nucleolus, worms with a disorganized nucleolus have overall higher activity levels. And this hyperactivity is reminiscent of brain hyperexcitability that occurs in ALS patients and causes muscle spasms, one of the earliest symptoms of disease. So in summary, we found that yes, nucleolar disorganization can cause ALS-like symptoms. And now we want to understand, how does this happen, and could targeting nucleolar organization represent a new therapeutic approach to treat ALS? And with that, I want to thank all the members of my lab at MDIBL who have contributed to this work, as well as others at the lab. And I want to especially thank my sources of funding from the National Institutes of Health.
Any of your samples of C. elegans present with mutations? And if so, are any of those mutations usable in your research?
So yes, we can have spontaneous mutations arise. And in fact, that's how a lot of development and how much we know about the nervous system came from a mutation that caused some kind of observable change in the worm. And then we can figure out what gene that mutation was in and what pathway that is. But most of the time, we are introducing mutations that might be found in ALS patients into our worms and then studying the pathogenesis.
So I saw one of the sources on the images was openworm.org, and C. elegans is really simple. Have you simulated a worm?
Have I simulated a worm?
What is openworm.org?
Openworm.org is a compilation of lots of literature and images and information about C. elegans. So the C. elegans community is like crazy collaborative. Maybe because it started off as a smaller community. Now there's thousands of labs across the world that use this animal to study biomedical research. But there's so many. There's Wormbook, Wormbase, openworm.org. Those are just a couple of the resources.
Go ahead.
So you say you edited its DNA, and you said you did this on a living worm. So what kind of tools do you use for that? Because in my head, I'm imagining opening up its genome like a Word doc and making edits. But I know that's not how it works.
That is a great way to think about it. Actually, a lot of times we think about it just like moving puzzle pieces around. So you may have heard of CRISPR DNA editing. So that's the technology that we're using in these worms. Worms are really amenable to that technology. So we inject all of these tools, an enzyme that cuts the worm's DNA, and then a piece of DNA that we want to put in there, like a piece of DNA that encodes the fluorescent tag, for example. We can put that wherever we want in the worm's DNA. We inject it into the reproductive system of the worm, and then each worm, they're hermaphrodites, so each worm can produce 300 identical copies of itself in 3 days. So we just screen their progeny for whether or not we were successful in making that edit.
So the organization of neurons in the human brain is obviously, and number is different from that in C. elegans. Could that create a problem in trying to move the information gotten from C. elegans into helping humans?
Yes, so C. elegans is an excellent tool for understanding basic biology that all animals share. And it turns out that a lot of the processes that are disrupted in disease are important for all animals, including the nucleolus. The nucleolus is an essential compartment of C. elegans cells and human cells. With that said, whatever basic biology we uncover in C. elegans, we'd absolutely need to follow that up in mice, probably, and then in humans. Yeah.
Do you have a hypothesis as to why the nucleolar disruption specifically causes increased brain activity?
So am I at a scientific conference here? Right? That's a good question. So I think that there is just a change in circuitry, a change in the balance between excitatory brain cells and inhibitory brain cells. I don't know. So the motor circuit in C. elegans, there's like 5 different cell types. So that's kind of in our future directions to narrow down what brain cell might be disrupted causing this hyperactivity. But I also want to say the hyperactivity is in young adult worms, so day one adults. We're really interested in looking at how those locomotion differences might change in aged worms, which is more relevant to these age-related diseases like ALS.
What do those poor little worms do in the real world?
So these are soil-dwelling nematodes, and they eat typically rotting fruit in the wild. We keep them in the lab in little petri dishes, and they eat a non-pathogenic form of bacteria, of E. coli. Yeah.
I think you're off.